Yeah, thank you very much, Barbara, Lynn, Philip, Blanca, everybody who is involved in that exciting project. I'm really happy to talk here. I might be a little bit of a host, but that doesn't mean that I'm less nervous. So I'm really happy to present some of my most recent research results, which I have published in a book. And you'll see that it is kind of a collection or an integration of neuroscience, a little bit of genetics, because that's my original background, neuroscience, and very much influenced by social work and social pedagogy, because these were people I started collaborating with, and they pushed me in a direction which I didn't expect before. And of course, overall framework, how could it be different than here at the KLI, is in some sort of evolutionary context. All right. Okay, so we're shifting a little bit of gear now. So we're talking about the most complex organ entity here, namely the human brain. The human brain is so complex, we're talking about 86 billion neurons. And these 86 billion neurons, actually, this is not on its own what makes the human brain so particularly or exceptionally powerful. It is the connections, how these neurons interact and communicate with each other. So we're talking here about the connection that lies somewhere in the range of 10 to the 14th to the 10 of the 15th synapses, synaptic connections. And in order to create such a marvelous organ, one would consider what is the evolution underlying behind it. So, I mean, what has made the human mind so powerful, so probably more powerful than, although that's debatable, than that of other animals? Well, if we look at the evolutionary trajectory or history, then we can see that especially over the last few million years, maybe the last 3 million years, with at least 2 big jumps, there has been an increase in the overall volume of the brain from around 500 gram in Australopithecus or in the very early ancestors of Homo habilis, up to the most recent Homo sapiens sapiens to 1.4, 1.5 kilogram of total brain volume. Of course, very much dependent on the size. Mine is much smaller, but I don't pity myself for that. So that's still okay. But as you can see, so we had a threefold increase in the overall amount of the volume of the brain. Now, especially the geneticists, and as I've already said, I've been trained in genetics. The endeavor was first to look at what are the genetic components or factors that have driven this huge expansion. So especially when looking at individual genes, there was the hope that we would find some kind of magical bullet that will give us an answer what sets the human brain apart from all other mammalian brains, say. And let me give you one of these examples, which I have been looking into a little bit more, actually driven by collaborations with an anthropologist, Dan Larson, and triggered by Werner Callebaut. So one of the genes I've been looking into, doesn't matter, was the SR-JAP gene. And this, thank you very much. And this SR-JAP gene has undergone very recently gene duplication. So we share that gene with our next relatives, primate relatives with orangutan chimpanzees. However, there were at least 2, probably 3 duplications. So, the most interesting one occurred about 2.4 million years ago. And that correlates roughly with the evolution from the Australopithecus into the hominin line. So that what you can see here is a fish staining. So it's on the chromosome 1, whereas this DNA sequence of the SR-GAP gene was marked. And in the human chromosomes, you can see already these two duplications that occurred at these time points of about 3.4 and 2.4 million years ago. So, right, that's very exciting. When we looked at that, we thought, that's brilliant. So we have something that might have been driven, this expansion of the human brain. Now, what is this gene we were looking at? So this is GTPase. For those of you who are a bit know a bit about molecular biology, then you will maybe know that this is a GTPase is very often involved in cell differentiation, in undergoing cell cycle, and so on. And in this case, it was particularly interesting because it has been shown through various experiments that it had some kind of function in, or was involved in neural migration, in neuronal differentiation, and what I was particularly interested in, this synaptic maturation. Now that was kind of the function of the gene, the wild-type gene itself. Now the duplication had a very different effect. In investigating that in heterologous expression systems and in culture, it could be has been shown by Sharia, but also by other groups, that the duplicated gene formed a homodimer with the wild type and exerted a dominant negative effect. So what happened then was it came to a downregulation of the effect of the wild type gene, and the normal function of the SR-GAP gene that usually is somehow involved in the neuronal migration, but in a way that it enhances the duration of the cell-cell contact, now has been overridden by these heterodimers, and it inhibited this cell-cell contact and made the neurons move faster along the radial glia cells. So that was already a really good indication. We thought, okay, why not? This might have driven expansion, the expansion of the cortex. On the other hand, equally interesting was that it was also shown that it had an influence and the maturation of the synaptic spines. So this is where the synapses between the neurons are built. These are very, on the dendrites of individual neurons, these are some specifications, some morphologies. And they, when these dimers were formed, then we had a delayed spine maturation and formation. So both findings extremely interesting. So the first one, very interesting because, as I said, we had this huge expansion of the human brain, especially the cortex was interesting. Because the cortex this is something that, particularly in humans and in primates, we have this 6-layered cortex in the brain. And this is something that is quite unique to primates. So it expands and it expands, but this clear 6-layered structure is typical for humans and our closest relatives as well. So this was the first thing we were really interested in. And the second thing, as I said, is this maturation of the spines. So here, this is a typical kind of probably a pyramidal neuron somewhere in the cortex, and this is how these specifications look at the dendrite. This is then where the synaptic communication would take place. This is a spine, and this is how an adult spine, or on the border between a adolescent rodent and an adult, this is how spines would look like. Now, if you do this experiment in mice and you co-express this duplicated SR-GAP2 gene, then you would get these immature spines at the same time point. But if you let it mature further, then it would create the normal spines as expected. So this was some good indications, and we were really interested. And we thought, okay, let's look at that. But as we've already heard in many talks today, that, and especially in that of Eva Jablonka, that, I mean, the genetic component is just one of the things. And what I've known from years and years before of the studies was that the phenotype of this delayed spine maturation, this is not something that is very unique to one gene. Actually, we see that in kind of many different types of mutations that are relevant for all sorts of brain pathologies. And this phenotype can be rescued with many different genes. However, very interesting is that this delayed maturation seems to be something that I would say is particularly prominent for that one gene that we were particularly interested. However, as I said, please keep in mind that spine maturation is in reality something very polygenetic, and we have a lot also of non-Mendelian contributions to that. And the same goes basically also for this expansion into the six-layered cortex, because even if we have had this one very exciting gene, of course, there's hundreds of groups, probably thousands of groups in neuroscience that are studying which genes could be involved. Maybe the most famous ones that came up in the last 15 years is these little mini brains that you might have seen in the Petri dishes. And they also looked at which genes are involved in making this multi-layered cortex. And in reality, there is, again, many more things than just like the one gene that we were interested in. In reality, we have something like genes that are involved in cell adhesion, in vesicle transport, and so on, but also in cell cycle regulation. So as you can already see, there is an exciting gene, and not just us, many people were happy to say, okay, this is a gene that might be really relevant, but you have to tone that down because there is many more to it than just that. And with that in mind, and of course, being in the environment where I am, we said, okay, let's also look at some other explanations that also contribute. So not just the gene or the function of the gene itself, but also from an evo-devo perspective. What does this function actually, or what happened then with the mutation because of the duplication? What happened then? How was the function changed? And what we investigated and saw, and I have to admit this is not the work that Dan Lars and I have been doing, but this was through literature research and so on, and communication with other researchers. So there was a clear heterochronic effect of this duplicated gene. What did it mean? So this is basically what I showed you before. So it was not that we had this gene effect and all of a sudden a complete new function has arisen, but what it did in reality was it changed the rate of the development by changing the overall duration of the maturation. So the spine, it took much longer for the spine to mature. So in the end, it was, as I said, not the magical bullet which just had one change in function. And this is one of the classical concepts in evo-devo, is that you actually see a heterochronic change, for example, and this gives you a new variation of features where natural selection can then act upon, right? So it's not so much about all these new functions. So then it's not all in the genes, or is it? And of course, I mean, as I said, when I originally studied, that was about at the end of the 80s, my professor then, Rudolf Schweyen, that some of you might still know, he kind of told me that there was about 100,000 genes in the human genome. And of course, now we know that, I started on yeast genetics, no neurons. Now we know we've got like something like 20 to 22,000 genes, right? That is just marginally more than Xenopithecus elegans, which is in itself already kind of a frustrating experience for a human being, right? But if you look at the figures from 22,000 to 86 billion of neurons, you can already see the gap, even if we include alternative splicing and differential regulation and so on. So how did this all come about? And again, here is where the KLI helped me in thinking about this concept. And this is something that I very much owe to Barbara Fischer, to Philipp Mitteröcker, and to Nicole Grunstra, because what they were doing was they were looking at different selective forces of newborn, when children were born. If I've used that picture that Barbara uses very often. So what you can see here is the birth channel of the mother, and this is the head of the newborn. In the case of the orangutan, the chimpanzee, the gorilla, and the human. And what you can already see, the human, here you have this very, very complicated and tight relationship. That means that giving birth in humans is extremely dangerous. So it's high risk because the head only just fits into the birth canal. And that has, for reasons there was, for that, this you should better ask others who are much more competent to talk about that. But this kind of obstetric dilemma, that there was this limitation in when a child, or in this limitation of how big the size of the head of a newborn can be, because, and also the underlying brain then, that led to the following observation, namely that human babies are born prematurely. Of course, we all know that when we've read Stephen Jay Gould, he calls the newborns, or the very young children, he calls them the extrauterine fetuses. So these children have a very long, so the human children, they are basically born with relatively immature brains. So Ladislav Kovac, in his book, a good friend of mine, he calculated and said, if we had humans born with the same cognitive abilities and capacities like a chimpanzee, they probably would have to stay in the human womb for about 20 months, which doesn't happen, obviously. So that means that human babies are born with relatively immature brains. And these immature brains, they develop outside, of course, the womb, which gives them many more stimuli and to shape the human brain. Now, at the same time, I've already said that we have we have a very long childhood and puberty, and this is something that you can always find in the literature as paramorphosis, which means that you have a delayed development of some parts of the human brain. And I showed you one of the genetic components that contributed to it. But of course, there's much more. And I mentioned that there are other genetic components. But what I would like to point out is that there is also much more environmental factors. And this is what I'm particularly interested in. And how did I get to this? Is because due to collaborations, communications with people from social work, we were looking at high risk and vulnerability factors in the brain development. Because during this very long childhood and puberty, the human brain undergoes a high amount of neuroplastic processes. So this is just one really nice example that shows you that the prefrontal cortex of the human brain, which is basically all that, but here is the region of interest highlighted, that this region, so this is color-coded, the bluer it is, the more mature the brain is, but this region matures until early adulthood, so probably until being at the age of 28 or something, or even more. And there is many, many more neuroplastic processes. I was, as I said, particularly interested in synaptogenesis and the pruning of synapses. How am I doing for? Okay. Mm-hmm. Now, this is again something that's not new. Actually, probably the most important neuroscientist, Santiago Ramón y Cajal, has already shown exactly that. But this has been forgotten and has only been revived in the last maybe 30 years. So what is shown here, these are slices of the human cortex. And what you can see already is that we have also the newborn has already quite a lot, probably as many as an adult, neurons present in these slices. So these are the neurons with the protrusions, which are probably the axons. But what you also see, that the connections, the synapses are not yet formed. So synaptogenesis has not yet occurred to that extent. And this is, as I said right at the beginning, that what I was particularly interested in, right? And then if you follow the developmental trajectory over the first 2.5 years, there's a huge increase. There is a synaptic exuberant growth of synapses that get then trimmed down. So from the age of 2 to our brains, we actually lose about half of the synapses again. This trimming down, the synaptic pruning occurs. I'm jumping in the interest of time. I'm jumping over my little movie. 10 minutes. Okay. All right. Then I give you the movie. All right. This is a heterologous expression of some components that create actually a synapse. And what you can see here is this is in a heterologous expression system a synapse that would be formed developmentally and stabilizes this connection, the communication between the neurons. If there is no synaptic connection for whatever reason, because there is developmental nothing that is that it can communicate, or because it had to it was trimmed down because of this use it or lose it principle, because of losing it in the sense of not being used, then you get a retraction of the filopodia. So this is what simply occurs when new synapses are formed, or if you lose the synapses. So I'm just highlighting that because I think it's nice. It illustrates what it means to have a stable synaptic connection and a retracted synapse. Of course, this is much, much more mobile, but this is just something that, because it was a live image, and I'm still emotionally attached to that film. So, but why do I show you that? Is because in order to stabilize these synapses, this is something that needs the input from the environment. This is not something, so you have this exuberant growth that is developmentally driven through genetic processes and so on, but to be stabilized, that pruning occurs, and only that remains what is relevant. This is due to the environmental inputs. So here you have neuroplasticity that builds a direct bridge between the environment and the individual development. Okay, not so much new yet, but it is still extremely important because we have this very, very long development, remember? And actually, neuroplasticity is something in humans, not in other mammals, but in humans, doesn't stop. So even until we die, there is, of course, to a different extent, but until we die, we have that. And so I just wanted to cite Quartz and Sainovsky here, who said that our human mind and this magnificent structure, including language, complex thinking, social skills, you need to build that by putting layer upon layer of experience. And this comes, on one hand, you have this organizational or hierarchical organization, namely the 6-layered construction, but this goes hand in hand with learning processes. And these learning processes are particularly prominent because of our protracted development. So here we have a long development that leads to the process where we have more and more paramorphic descendants that evolve, right? So we have an increase in more and more offspring that has this extended long development. And this makes us a paramorphocline species. I'm very happy to discuss that if this is really true, but this is the underlying argument now, or a hypothesis. Now, how does this come about? And I'm not need to go into too much detail, but because I think here you're most of you are familiar with that. So all these learning processes that are particularly effective in early childhood, because we have such a high neuroplasticity, are due to being socially embedded, so that human children do not only have so they have the learning processes that come directly from the parent-offspring communication, and but they also have these social learning processes that come from alloparents, like grandmothers. You're probably all familiar with the grandmother hypothesis. So here we have this really strong effect on crosstalk between our cultural environment, especially the social cultural environment, and brain development. Now, that means that our alloparents provide some sort of scaffolds. So they give us something to develop along, right? These are just examples of scaffolds, like imitation, social referencing, joint attention, reciprocal interactions, all that. If any of that does not work, you can actually see that in children who have deficits there. And this is what I've been pointed to. So rather than having just this genetic inheritance on one hand of the organism, and then you have, on the other hand, these resources that are relatively passive in the environment and just exert natural selection pressure via natural selection, what you actually have is you have much, much more. You have the genetic and the epigenetic inheritance, which Eva Jablonka talked about. You have also niche construction that humans create the environment they live in by reciprocal interaction, by including scaffolding, and so on. And these constructed niches, we transmit to our children, namely not only the ecological, cultural resources, but also the social ones. And why is this relevant? Because this is something that you probably have come across already. Depending on the environment you're living in, this was in mouse studies, but I mean, in correlation studies, it has been shown for children for quite a long time. The more impoverished the environment is, the less arborization you have in your neurons, the less dendrites and dendritic spines you have in your neurons, and the overall amount of the volume of the hippocampus, for example, goes down. I jump over that because I think I don't have much time. How much more? 3 minutes, okay. Now, why is this relevant? Because the social communications or the social interactions with the parents or alloparents and the child, they are particularly important in early infancy because at that time, children cannot regulate their own stress axis. So children have not yet a fully developed stress axis. They can produce high levels of cortisol, but what they cannot produce is, or they cannot downregulate it. And actually, this is something that already happens very early, so can already happen in the womb when mothers are exposed to high stress levels. Then only if the cortisol is very high, usually it doesn't happen, but when the cortisol is very high, then this can be transmitted to the child, to the fetus, and the fetus then is already born in the hippocampus with a reduced amount of glucocorticoid receptors. And that means that the overall possibility of downregulation of the stress axis is not provided anymore. They cannot regulate it anymore. And this is even more important because don't forget that if it's a female fetus, then the oocytes are already generated in the 4th to 5th months of the gestation age, right? So this is, of course, something that you then also already transmit at least 2 generations. And let me just close up with that with that slide. So if you if you study stress on its own, then you will already see, and this has also been shown, that it affects not only the morphology of the neurons, but also the size of the hippocampus, and this would then lead to cognitive impairments in rodents, in other animals, and also in humans. This has been in humans, as I said, in correlation studies. But what's new or relatively new is that if this maltreatment, abuse comes due to social interactions, because it's exerted by the mother animal in this case, then it could be shown that this causes an early maturation of the amygdala. The amygdala is the organ that is involved in anxiety and fear. And why is this relevant? Because at the beginning, children only have preference learning, and we know that because of the maturation of the amygdala happens at the same time, kind of, when aversion learning sets in. This is when children get this stranger anxiety at the age of 7 to 8 months. So this means when the maltreatment comes from the mother, this sets in much earlier than normally. So here you have again a shift in the developmental trajectory. So let me just say that we have in brain development much more than just the genetic factors, and I believe that they will also be relevant for transgenerational effects. Yeah, I'm just citing Eva Jablonka's 4-dimension of transgenerational inheritance. And with that, I would like to say thank you for activating your neuronal assemblies.